स्टार्ट करें. Good afternoon and welcome everyone to this KVPY previous year question discussion session. And today we are going to discuss the questions of 2018. Great. So precisely KVPY SA 2018 paper, the biology sections, both the part 1 and part 2 of biology. Great. Okay. So without wasting a lot of time, let's start that. The first question of part 1 biology is, which one of the following molecule is a secondary metabolite? Right? So what should be the correct answer? Karthik, Akhil, Abhivir, please attempt this question and let me know what should be the correct answer here. Which one of the following molecule is a secondary metabolite? Which of it? Abhivir is saying C, penicillin. What about others? Okay, Agrim C. Okay. Great. So the correct option here is C only, right? So correct option is C. Penicillin is the correct answer for this question. Now, why so? Let me brief about this. See, in any living organism, there is metabolism. Okay? And when metabolism happens, there are a lot of chemical molecules that are being produced, right? So this metabolism results in the production of molecules called metabolites, okay? Or all the chemical molecules that participate in a metabolic reaction are called metabolites. Now these metabolites are of two types, okay? What are these? These are the primary metabolites and the secondary metabolites, okay? Now let me tell you, the primary metabolites are the one which are participating in the primary metabolism, okay, that is the metabolism which is crucial for the, for the life of the organism, without which the organism will not survive, it will die. But the secondary metabolism, this is not so. The secondary metabolism are the metabolic reactions which provide certain advantage. They are not crucial for the living, but they provide certain survival advantage. तो यहाँ पे ध्यान रखिए, this is crucial for living, okay? So absolutely necessary, but this is not crucial. This is not crucial for living, but provide survival advantage, okay? समझ में आ रहा है? Now see, ethanol, it is primary for all the organisms who are anaerobic and they produce ethanol, alcoholic fermentation. Great. Lactate, again, it is primary metabolism. Why? Because it is the product of, again, the lactic fermentation. Respiration, which is primary metabolism. Penicillin is a molecule which is a product of secondary metabolism, and it is not crucial for living. It just provides survival advantage because it will kill the other microorganisms nearby, okay? And thus it is going to help the organism to get maximum of the natural resources in the nearby environment. समझ में आ रहा है? So जो आपका पेनिसिलिन है वो एक तरह का एंटीबायोटिक है, आप लोग सब जानते हो, और ये different जो अदर लिविंग ऑर्गेनिजम्स हैं इन द नियर बाई सराउंडिंग उसको किल कर देता है, सो दैट द रिसोर्सेज आर अवेलेबल फॉर दिस पर्टिकुलर माइक्रोब एंड नॉट फॉर द अदर्स, ओके? सो इट्स अ काइंड ऑफ competitive advantage. And then citric acid, again citric acid is produced during the Krebs cycle, part of aerobic respiration, the primary metabolism. Great. I hope it is clear what is primary and secondary metabolite. So let's move on to the next question. Okay, next question. So what is the next question here? Next question was, lecithin is a carbohydrate, phospholipid, nucleoside, or protein? What should be the correct answer? What should be the correct answer? Okay, Akhil is saying B, nucleoside, no, sorry, phospholipid. What about others? What a lecithin is? Is it a carbohydrate? Is it a phospholipid, a nucleoside, or a protein? Okay, so Akhil is saying phospholipid, and that is the correct option here. It is a phospholipid. How it is? See what lecithin is. If we are looking at lecithin, lecithin is chemically known as DPPC. DPPC. Okay, that is, the full form for this is dipalmitoyl phosphatidyl choline. Okay, this is the actual molecule. The chemical name for this molecule is DPPC, that is dipalmitoyl phosphatidyl choline. Di का मतलब क्या है? Two. Palmitoyl is for palmitic acid. Okay, phosphatidyl because it has phosphate group. Okay, and choline because it has choline sugar. Sorry, choline alcohol. Okay, so this particular molecule is going to look something like this. So it has a glycerol. Okay, it has 3 OH groups. The first OH, second OH, and third OH I am making here. Okay, now with the first and second OH, there are 2 fatty acids. Here the palmitic acids are attached. Okay, with the ester bond, and with the third OH there is a phosphate group, and with this phosphate group there is choline attached. समझ में आ रहा है? So this is how this lecithin looks like. Lecithin is a what? Lipid. It is a lipid. In fact, it is the most common lipid in animal cell membrane. Okay, and here when we are talking about the animal cell membrane, we mostly mean the Donan's membrane, which is the cell membrane of RBC. किसका? RBC का. समझ में आ रहा है? तो जब भी हम लोग मॉडल एनिमल मेम्ब्रेन की बात करते हैं, बेसिकली वी आर टॉकिंग ऑफ द RBC membrane, that is the Donan's membrane. Okay, and this is what DPPC is. That is lecithin, so it is a phospholipid because it has phosphate group. ठीक है? Now, if we have to see the chemical structure of it, this is how it looks. देखो यहाँ पे. So this particular chain, this molecule is glycerol. It has 3 OH here. Okay, and with this OH, the 2 OH, there is 1 palmitic acid. This is another palmitic acid, the fatty acids. And then if you see closely, here is the phosphate. And then lastly, here you have the choline group. Got it? So this is what? Lecithin or DPPC. Now, let me tell you one more thing here. If you look this portion of this lipid, this is polar. This is polar, so it is hydrophilic. And if you look at this part of lecithin, this is non-polar, okay, and hence it is hydrophobic. It is hydrophobic. Got it? So this lipid molecules have both hydrophilic and hydrophobic part. This hydrophilic part is also known as head, okay, and this is known as tail. समझ में आ रहा है? Since they have both hydrophilic and hydrophobic, we call them amphiphilic molecules. These are amphiphilic molecules and hence they act as detergents. ये किस तरह काम करते हैं? Detergents की तरह. Great? Yes. Akhil, not lipophilic. Okay, not litho. Litho का मतलब कुछ और होता है. ठीक है? अगर आप litho लगाओगे, it means it is inorganic. Okay? So it should be lipophilic or hydrophobic. ठीक है? Not litho. Just pay attention to your spellings there. Great? Okay. So this was the second question. Let's move to the third question. Okay? What is the third question? The third question is the water potential, right? Okay. So the water potential of pure water at standard temperature and atmospheric pressure. यहाँ पे ना question में at रह गया है. So add कर लीजिए. The water potential of pure water at standard temperature and atmospheric pressure is, okay, A 0, B 0.5, C 1 and D 2. Now this is a very fundamental question है ना, इसके लिए आपको किसी calculation को करने की जरूरत नहीं है. Okay, it's very fundamental. Now you know, yes, I can see the comments of Akhil and Agrim. Both are saying A. Great. That is 0. And you both are correct. Okay. Yeah, Avivir, correct. Very good. So 0 is the correct answer. है ना? The water potential of pure water at standard pressure and temperature is 0. It is 0. Okay. So psi p of pure water is 0 at STP. ठीक है? इसका मतलब क्या होगा? एक और चीज आप देख लो. यहाँ पे psi p लिख दिया गया है, लेकिन वाटर पोटेंशियल को यूजुअली हम लोग psi w से डिनोट करते हैं. Okay, and there is a equation psi w is equals to psi p plus psi s. This is pressure potential, this is solute potential, and this is water potential. Now let me tell you, solute potential is always negative. It is always negative. इसका मतलब अगर आप कोई solution लोगे, right? कोई भी solution लोगे, not pure water, कोई भी solution होगा, मान लो psi p at standard, मतलब at STP it is equals to 1. So मैं इसको सीधा 1 ले रहा हूँ. Okay? psi p को मैं 1 ले रहा हूँ. Plus psi s कुछ भी क्या हो जाएगा? Okay? अगर कुछ भी आप इसमें solute डालोगे, it will be negative x, and therefore always this psi w will be negative for a solution at STP. है ना? याद रखिएगा. For a solution at STP, the water potential will always be negative. Okay? Only for pure water at STP it is 0. Great, good to see कि आप लोगों को याद है ये. Very good. Let's move to the question number 4. Right? The question number 4 says action potential, right, in neurons is generated by a rapid influx of. What should be the answer? Fatafat, attempt कीजिए इस question को. The options are chloride ions, potassium ions, calcium ions, or sodium ions. What should be the answer? What should be the answer? Okay. Agrim is saying D. Akhil is saying D. What about others? फटाफट comment कीजिए. See, when we are talking about action potential, okay, action potential, देखो, normally in a nerve cell, there is a negative charge on the inner side, that is in the cytoplasm, and positive outside. Okay? If by any chance this changes, this charge is reversed, that is negative outside and positive inside, okay, then such a condition is called depolarized state. It is called depolarized state, okay, and this is only what we understand as impulse. Okay, this is what an impulse is, और impulse को ही हम लोग क्या बोल देते हैं? Action potential. Okay, now if we look at the first initial state where the cytoplasm is negatively charged and outside is positive, this is called polarized state. This is called polarized state. Okay, and this is the resting condition. This is the resting condition. Okay, now in order for this to happen, okay, in order to this to happen, that is the switching of polarity from negative to positive, what is required is the opening of sodium channels here. Okay, so there are these sodium channels, okay, and when they open, on the outer side there is a lot of sodium ions, okay, on the inner side the sodium ions are less. There are less. So as per concentration, this sodium rushes in. When this sodium channel opens, okay, the sodium ions rushes in, leading to the switching of the polarity from negative to positive. That leads to generation of this impulse or action potential. Okay, got it? So the correct answer is D. Congratulations, you all are correct. Great. Okay. Now moving on to the next question. Question number 5. See, this question is erythropoietin produced by, and the options are heart, kidney, bone marrow or adrenal gland. Now फटाफट इसको attempt कीजिए. Let me see. Okay, great. So Akhil and Agrim are saying B, that is kidney. They are saying B, that is kidney. Let's see. First of all, your answer is correct. Okay, kidney is the right answer here. Now, what happens and how it happens, let me tell you. See, the formation of blood is called hemopoiesis. Okay, that is called hemopoiesis. Hemopoiesis. Formation of RBC is called erythropoiesis. Okay, it is called erythropoiesis, and erythropoiesis, okay, erythropoiesis, wherever the organ in which erythropoiesis happens is called erythropoietic organ. It is called erythropoietic organ, and within erythropoietic organ, the erythropoiesis happens because of a compound called erythropoietin. Okay. So this is the erythropoietin that induces erythropoiesis. Okay, so erythropoiesis is induced by erythropoietin. Now this erythropoietin is secreted by, it is secreted by kidney. Okay, now within kidney there are some special cells. Okay, these cells are called interstitial fibroblasts. Okay, these are called interstitial fibroblasts that secrete this erythropoietin, and these cells are found in the JGA, that is juxtaglomerular apparatus. Great. तो थोड़ा सा आप लोग ध्यान रखोगे कि within the kidney there are nephrons, and near to the glomerulus, near to this renal corpuscle, there is a special arrangement of cells, okay, at the junction of efferent arteriole, DCT, and the glomerulus. This forms JGA, juxtaglomerular apparatus, and it is endocrine in function. It releases a hormone called renin, as well as a particular molecule called erythropoietin, which is responsible for erythropoiesis, that is formation of blood. Great. याद रखिएगा ये कब होता है? Whenever your body senses that the volume of blood is less, okay, or there is, you know, decrease in the blood pressure, then this JGA will produce erythropoietin, inducing the formation of new blood cells, so that overall blood volume can increase back to normal blood volume and blood pressure. Okay, great. So this is it. Great, good to see that you all remember these things. Let's move to the next question. So the next question is, tendrils are modification of, fatafat. See, tendrils are modification of what? Tendrils जो है ना वो कुछ ऐसी चीजें होती हैं which helps in climbing. Okay, and these can be modification of terminal leaves, okay, or even for the stem. Okay, ऐसा भी हो सकता है, या फिर ऐसा भी हो सकता है कि लीफ का जो टर्मिनल है वो पूरा मॉडिफाई हो जाए, या फिर ऐसा भी हो सकता है कि यहाँ पे लीफ है और यहाँ पे जो आपका स्टेम होना चाहिए था वो किस में हो जाए? टेंड्रिल में हो जाए. So anything can be possible. Great. So the correct option is obviously option number A. Good to see that again. All of you all are correct. Stem or leaf both can modify into tendril. Okay, and there are different portions of stem and leaf that can modify into tendrils. Great. Let's move on to the next question. So next question is question number 7, and this question says, which one of the following combination of biomolecules is present in the ribosome? क्या होगा सही आंसर? फटाफट. What should be the correct option here? Okay, Akhil is saying C. Okay, that is RNA and proteins. What about others? फटाफट comment कीजिए. So when we are talking of ribosomes, ribosomes are of two types, 70S, 80S, and every type of ribosomes has two subunits, a smaller subunit and the larger subunit, right? In case of 70S, which is prokaryotic, okay, the two subunits, smaller subunit is 30S, the larger subunit is 50S. Now, this is made up of 16S rRNA and proteins, right? When we are talking of 50S, it is made up of 23S rRNA and 5S rRNA and proteins. है ना, थोड़ा सा ध्यान रखेंगे आप लोग. When we are talking of 80S, again it has two subunits. The smaller subunit is 40S, larger subunit is 60S. 40S is made up of 18S rRNA and proteins, okay, while 60S rRNA is made up of 28S rRNA, 5.8S rRNA, and 5S rRNA, plus proteins. Okay, so no matter which ribosome you consider, every ribosomal subunit is composed of RNA and proteins, and particularly which RNA it is rRNA. rRNA का full form क्या होता है? Ribosomal RNA. Okay, and let me tell you one more thing. In these two, this 23S rRNA and 28S rRNA are not only structural, but they also have enzymatic property, that is catalytic property, है ना? इसीलिए हम इसको राइबोजाइम भी बोलते हैं, याद रखिएगा. What we called राइबोजाइम. Similarly, इसको भी हम क्या बोलते हैं? राइबोजाइम. Okay, great. Okay, I'm so happy to see आप सब लोगों को याद है ये सब कुछ. Great. Question number 8. Which one of the following proteins does not play a role in a skeletal muscle contraction? What is the correct option here? Akhil is saying D, microtubule. Correct option. Please attempt to this question. Fatafat. See, when we look at muscle, right? So most of you are saying D, that is microtubule, and it is correct option. So option D is the correct answer. Okay. So D is the correct answer. A microtubule. Rest all of these are part of sarcomere, है ना? Actin, myosin, troponin, and troponin के साथ एक और चीज होता है tropomyosin. Okay, so these four are the part of sarcomere mainly, ठीक है? And they thus help in muscle contraction. ठीक है? हम लोग एक काम करते हैं, फटाफट देखते हैं थोड़ा सा. I'm going to show you some images here. So this is image from NCERT. This is a characteristic TEM image showing the striated muscle. So you can see here the dark band, जिसको कि हम लोग A band बोलते हैं, anisotropic band, and this is the I band, that is isotropic band. It has only actin plus tropomyosin plus troponin, while here you have myosin. Okay, you can see this one sarcomere here. So this is myosin here, and these are actin fibers. Okay, if we look at more, if we try to have a more closer look on the actin, so see, this is actin. Okay, and now actin, on one actin there are these two molecules of tropomyosin. Can you see this tropomyosin? And at frequent interval there is again another complex protein called troponin. Great. So just take a glimpse of it. Okay, great. Moving on to the next question. Question number 9. Which one of the following reactions is catalyzed by high energy ultraviolet radiation in the stratosphere? क्या होगा? Option A, O2 plus O, O3, that is ozone. O2 dissociating into two nascent oxygen atoms. O3 plus O3 combining to form three O2 molecules. Or option D is O plus OO2. Now, which of these reactions is catalyzed by the UV rays in the stratosphere? Dekho, if we are talking about stratosphere here now, so stratosphere, upper stratosphere has ozone layer. Why? Because it has a high concentration of O3 gas, okay? O3 gas, ozone gas. Correct? And what this ozone gas does, it absorbs or reflects. दोनों काम करता है, absorb भी करता है, reflect भी करता है. Most of the UV light coming from sun. right? Thus helping or saving the life on Earth from this harmful UV radiations, which are a strong mutagens. They cause mutation. Now, in the stratosphere or in the ozone layer, they do two kind of reactions, okay? They do two kind of reactions. What they do, O2 plus O, they convert into O3, okay? At the same time, they are responsible for decomposition of O3 into O2 plus O. So both of these reactions, okay, is carried out by this UV rays. Okay? So UV rays can carry out this reaction. Great. So what should be the correct option here? A. Okay, ध्यान रखना आप लोग इसका. A should be the correct option here. Okay, O2 plus O giving rise to O3. At the same time, decomposition of O3, that is ozone, into O2 plus nascent oxygen. Both of this reaction is carried out by the UV radiations. Okay, so there is dynamic things going on, and how much ozone will be there, this depends on the various abiotic factors like temperature and wind current. Okay, great. So please make a note of it. Moving on to the next question. Question number 10. Now, this question is, which one of the following statements is true about trypsinogen? Okay, trypsinogen. See, when we are talking of trypsinogen, trypsinogen is a protease. Trypsinogen क्या होता है? It is a protease. लेकिन ये कैसा है? ट्रिप्सिनोजेन जो है ना, ये एक इनएक्टिव फॉर्म है. ये कैसा है? इनएक्टिव एंजाइम है. और इनएक्टिव एंजाइम को हम लोग क्या बोलते हैं? जाइमोजेन. सो इट इज अ जाइमोजेन. ओके, नाउ दिस trypsinogen, which is produced by pancreas, is converted into its active form called trypsin. Okay, this is active. This was inactive. By which one of the following? Enterokinase, renin, pepsin, or it does not need activation? ये option तो बिल्कुल ही rule out हो गया, right? It requires activation. Okay, so I can see Akhil, Agrim, both of you are saying A, that is enterokinase, and you all are absolutely correct. Enterokinase. Okay, so enterokinase is responsible for conversion of trypsinogen into trypsin, and then only it can act on the proteins for its digestion. Now this enterokinase is secreted by, कौन secrete करता है? It is secreted by duodenal mucosa, है ना? यानी कि duodenum का जो mucosa है वो secrete करता है किसको? Enterokinase को. ये चीज again आप लोग ध्यान रखोगे. This may be asked in future papers. ठीक है? So just keep a note of it. Great. Mucosa. Okay, great. Moving on to the next question. This is question number 11. Which one of the following organisms respire through skin? Okay, respire through skin. Let's see. Now, if we look at this question, basically we want to know that which of these organisms have or show cutaneous respiration. है ना? Akhil, don't put a question mark. Give me answer. Right? See, respiration by skin is called cutaneous. Okay? Now, let me tell you a few more things. देखो, when we are talking about blue whale, है ना? So whale is a mammal. Is a mammal, and all the mammal shows respiration by lungs. और lungs respiration को हम लोग क्या बोलते हैं? Pulmonary respiration. We call it pulmonary respiration. A. B is salamander. Okay? Salamander. Salamander is a member of amphibia. Yes, correct, Sushweta. So just by elimination you can work it out. But let me tell you in detail here. So salamander is an amphibia and they respire through their skin, not by lungs, skin. So it is cutaneous. समझ में आ रहा है? So answer क्या होगा? Salamander. लेकिन हम लोग बाकी options भी देख लेते हैं. Platypus. Duck-billed platypus. आप लोगों ने सुन रखा होगा. Correct? It is what? A mammal. It is again a mammal. Duck-billed platypus. What about D? D is peacock. Peacock obviously is a bird. और bird में भी कैसा होता है? By lungs. So it is also pulmonary. ठीक है? So the correct option is salamander. Okay? Great. Let's move to the next question. Question number 12. Now this question says, which one of the following human cells lack a nucleus? Now very easy question. Great. Perry the platypus. Okay. What should be this? The answer of this question? फटाफट tackle करो जरा. बहुत ही easy question है. Which one of the following cell lacks a nucleus? So basically, which is enucleated, right? The option is C. Matured erythrocytes, right? बचपन से पढ़ते आ रहे हैं हम सब लोग. Matured erythrocyte does not have nucleus. However, immature erythrocytes do have nucleus, right? तो अगर ये question immature का होता तो ये ऑप्शन नहीं होता. ठीक है? Immature RBC have nucleus. Only during maturation they lose it. Great. Okay. Okay. Let's move on to the next question. Question number 13. Now this question says, the first enzyme that the food encounters in human digestive system is, now it is a tricky question, है ना? आपको आता भी होगा, लेकिन you may fall in trap of this question. A is pepsin, B is trypsin, C chymotrypsin and D is amylase. है ना? क्या होगा इसका आंसर? अच्छा, Akhil is asking, sir, what is keratinocytes? बेटा keratinocytes जो होते हैं ना, these are the cells which have epithelial cells which have deposition of keratin, okay, and because of this high deposition of keratin, they ultimately dies and forms the upper dead layer of your skin. ठीक है? तो आपके skin के जो cells होते हैं, upper dead layers which have high percentage of keratin are known as keratinocytes. These are kind of simple, these are compound epithelial cells. Okay? Okay, great. अच्छा, यहाँ पे आप लोग आंसर बोल रहे हैं D, right? Yes, so you all are correct. D is the correct option. Why? Because digestion in human digestive system starts from your buccal cavity with the help of salivary amylase, that is also known as ptyalin, right? तो ये किस एंजाइम की बात कर रहा है? ये बात कर रहा है salivary amylase की, okay? जिसको हम लोग commonly क्या बोलते हैं? Ptyalin enzyme. Great, very good to see that. Okay, so let's move to question number 14. Now, this question says, glycoproteins are formed in which one of the following organelles? Okay, glycoproteins are formed in which one of the following organelles? The options are peroxisomes, lysosomes, Golgi apparatus, or mitochondria. What should be the correct answer here? Akhil is saying C. What about others? See, देखो, peroxisome, it is mainly associated with the oxidation reduction reaction, okay? Lysosome, it has hydrolytic enzymes, so associated with the digestion or intracellular digestion. Golgi apparatus, इसके बारे में बाद में बात करता हूँ. Mitochondria, it is powerhouse of the cell, right? So it is mainly associated with the generation of ATP, correct? So if you do elimination, you come up to C. However, we are not going to eliminate here. C, Golgi apparatus receives the secretion or the materials from both RER and SER, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, you already know that rough endoplasmic reticulum is responsible for the metabolism of protein, protein production, protein folding, protein storage, correct? And SER for the production of lipid and carbohydrate. Okay? Now, what is glycoprotein? If we are talking about glycoprotein, it is a hybrid molecule which has two parts to it. One is glyco and second one is protein. Glyco का मतलब होता है carbohydrate and protein is obviously protein. Okay, it means this is a molecule which has two parts. One part is carbohydrate, the other one is protein. कहाँ से बनेगा ये? Right? ये कहाँ बनेगा? Only in the Golgi apparatus. Why? Because this carbohydrate is coming from SER and protein is coming from RER. Now both in GC, Golgi apparatus, will become glycoprotein. Okay? And this process is called glycosylation. This process is called glycosylation. Okay? Akhil, peroxisomes are not F0 F1 particle. ये मत सोचिएगा आप. F0 F1 particle का दूसरा नाम is oxysome. Peroxisome are cell organelles. They are individual cell organelles, okay, which are responsible for the productions and decomposition of peroxides like H2O2. Okay, peroxides वाला काम करते हैं वो. Okay, F0 F1 particle को हम क्या बोलते हैं? Oxysomes. Okay, great. Okay, so let's move on to the next question, question number 15. Now this question says, an example of nastic movement, that is external stimulus dependent movement in plants is, okay, the options here are folding up of the leaves of Mimosa pudica. Mimosa pudica, इसको हम लोग commonly बोलते हैं touch me not plant. है ना? It is touch me not plant. B is climbing of tendrils, growth of roots from seeds, and option D is growth of pollen tube towards the ovule, right? Okay, Akhil, Agrim, they are saying option A, that is folding up of leaves of Mimosa pudica. देखो, इस question में ना थोड़ा सा अच्छा, Shreya is saying C, okay, okay, C, that is growth of root from seeds. Okay, let me tell you few some information regarding this. है ना, फटाफट हम लोग पहले देख लेते हैं and then we will see. देखो, when we are talking about plant movement now, it is mainly of two types. One is called tropic movement, and the other one is called nastic movement. ठीक है? Tropic movement, it is dependent on the direction of stimulus. Okay? And when we are talking about the nastic movement, it is independent of the direction of the stimulus. कहने का मतलब क्या है? कहने का मतलब ये है कि अगर हम ट्रॉपिक मूवमेंट की बात कर रहे हैं तो ये मूवमेंट या तो इन द डायरेक्शन और इन द ऑपोजिट डायरेक्शन ऑफ द स्टिमुलस होगा. ओके, मतलब मूवमेंट किधर होगा? द डायरेक्शन ऑफ द मूवमेंट इज डिपेंडेंट ऑन द डायरेक्शन ऑफ द स्टिमुलस. तो ऐसे मूवमेंट को हम क्या बोलते हैं? ट्रॉपिक मूवमेंट. ठीक है? यस, है ना? ये 10th में था आपके कंट्रोल एंड कोऑर्डिनेशन चैप्टर में. वेरी गुड, इफ यू रिमेंबर इट, नथिंग लाइक दैट. नास्टिक मूवमेंट क्या है? इट इज इंडिपेंडेंट ऑफ द डायरेक्शन, है ना? तो स्टिमुलस किसी भी डायरेक्शन से है, मूवमेंट जहां होना है वहीं होगा, उसी डायरेक्शन में होगा. इट इज नॉट गोइंग टू चेंज. ठीक है? नाउ लेट अस सी दीज ऑप्शंस, है ना? When we are talking of the pollen tube movement towards ovule, now it is dependent on the direction from where the chemicals are coming. Okay? The ovule releases some chemicals, okay, and from where these chemicals are coming, the pollen tube is going to move in that direction only. So the direction of movement is dependent on this external stimulus called chemical. इसीलिए हम इसको क्या बोलते हैं? Chemotropic movement. So this is an example of chemotropic movement. What about roots from the seeds? They always move towards the water or towards the ground. अगर water के terms में बात करें, then it becomes hydrotropic. और अगर gravity के terms में बात करें, then it becomes geotropic. What about tendrils? This always is in the direction of the tactile response. जहाँ पे भी substrate का touch होगा, उधर ही ये move करेगा. And this is called thigmotropic movement. This is called thigmotropic. Leaves of Mimosa pudica, the movement is independent of direction, right? So it is an example of what? Nastic movement. So correct answer is A. Correct answer is A. So all those who have given me answer A are correct. है ना? Rest of you, please look at your concepts. Question number 16. Now see, this question is, if the genotype determining the blood group of a couple is IA, I naught, okay, and the second parent is IA, IB, right? Then the probability of their first child having type O blood is, Acha, Akhil is saying A, Agrim is saying A, what about others? देखो, जब भी ऐसे क्वेश्चंस होते हैं ना, तो आपको करना क्या है? You have to first make the gametes. ठीक है, फटाफट हम लोग गैमेट बनाते हैं. ये जो फर्स्ट पेरेंट है, इसके दो गैमेट्स बनेंगे. एक होगा IA और दूसरा होगा I naught. इसके भी दो गैमेट्स बनेंगे, IA, IB, right? Now this gamete can fuse with the either of the two gametes. तो ये अगर इससे फ्यूज करेगा तो क्या बनेगा? क्या बनेगा? IA, अगर इससे फ्यूज किया तो IA, IB, अगर ये इससे फ्यूज किया तो IA, I naught, और इससे किया तो IB, I naught. ये क्या होगा? A. This will be AB. This is A and this is B, right? So their children can either have A, B or AB blood group. ठीक है? O blood group तो कहीं आ ही नहीं रहा ना, तो probability क्या होगा? 0. Correct? Very good. Directly हो जाएगा. Very good. Okay. Question number 18. If the H plus concentration of an aqueous solution is 0.001 molar, then the pOH of the solution would be. Now chemistry से भी ये question कर सकते हैं, physical chemistry, है ना? फटाफट करिए जरा. D. Akhil is saying D, 11. What about others? What about others? जल्दी कीजिए. Okay, Agrim is also saying D. Shreya is saying A. Avivir D. Okay. Oh, is it? Maybe, unfortunately, I skipped one question, so we will get back to 17. Don't worry. Just a minute. Is it 17 we have not done? Oh, okay. Anyway, we are doing 18 first, and then we will get back to question number 17. Okay. D. So all of you who are saying D, D is the correct answer here. See, you know, H plus and OH, है ना? If you look at the H plus concentration and OH, that should finally add up for a solution to 14, right? It should add up to 14. ये तो पता है आप लोगों को? Correct? है ना? Now, if we look at the H plus concentration here is 0.001, so it is like 3. करेक्ट, निकाल लेना आप नेगेटिव लॉग से, है ना, 3. So 14 minus 3 will be what? 11. So POH will be 11. ठीक है, सीधा सा है ये, ओके, सीधे सीधे निकलने वाला है ये. So the POH value should be 11. Great. Okay. So unfortunately I skipped this question. We are going back to this question number 17. Now this question is, a cross was carried out between two individual heterozygous of two pairs of gene, okay, assuming segregation and independent assortment, the number of different genotypes and phenotypes obtained respectively would be, है ना. So basically ये कह क्या रहा है कि हमने एक क्रॉस किया है between two individuals, है ना, and both are heterozygous for two pairs of the gene. ठीक है, उसके बीच में कैरी आउट किया, करेक्ट. सपोज कर लो कि हमने ले लिया यहाँ पे two individuals, individual 1 and individual 2, ओके. यहाँ पे जो हमने जीन लिया है सेम मेंडल वाला जीन हम लोग ले लेते हैं, कौन सा, RRYY, ओके. दोनों हेट्रोजाइगस हैं दोनों जीन के लिए, so both individuals are heterozygous for both the genes, correct. Now they are asking कि इनके जो प्रोजेनीज होंगे F2 generation, है ना, ये F1 generation होता है because hybrids are always F1 generation, okay. What should be the ratio of their genotypes and phenotypes, okay. Now if you remember the dihybrid cross, great, if you remember the dihybrid cross, you know that we are going to get genotype in this ratio, 1:2:1:2:4:2:1:1, correct. So how many different genotypes are formed here? 1, 2, 3, 4, 5, 6, 7, 8 and 9. How many phenotypes will be formed? It will be 9:3:3:1. 9 round and yellow, round and green, wrinkled and yellow, and this is what? Wrinkled and green. So 4 different type of phenotypes, correct. So what should be the correct option here? Option C, 9 and 4. So in a dihybrid cross, in F2 generation, 9 different type of genotypes and 4 different types of phenotypes are formed, correct. Given that there is segregation and independent assortment of the alleles, okay. अगर यहाँ पे segregation और independent assortment नहीं होगा, मान लो कि ये linkage का केस होगा, तो ऐसा हो भी सकता है और नहीं भी हो सकता है, ठीक है, ओके. क्योंकि लिंकेज भी दो तरह का होता है, कंप्लीट लिंकेज एंड इनकम्प्लीट लिंकेज. तो इनकम्प्लीट के लिंकेज के केस में ऐसा हो भी सकता है, कंप्लीट लिंकेज के केस में तो बिल्कुल नहीं होगा, ओके. ग्रेट, इज इट क्लियर? I can see most of you have given the right answer. So good enough. Let's move to question number 19. Now this question says, consider the following vision defects listed in column 1 and 2 and the corrective measure in column 3. Choose the correct combination. What should be the correct answer for this? फटाफट आप लोग बताएंगे मुझे जरा क्या होना चाहिए? Akhil is saying B. Okay. You are saying B. What about others? Okay. Sushweta is saying B. 2. Okay. देखो यहाँ पे ना एक चीज फटाफट देख लो. हाइपरमेट्रोपिया को हम लोग बोलते हैं फार साइटेडनेस. हाइपरमेट्रोपिया को क्या बोलते हैं? फार साइटेडनेस. एंड इन ऑर्डर टू करेक्ट दिस डिफेक्ट वी यूज convex lens. We use convex lens. Okay. If we are talking about myopia, myopia is what? Nearsightedness. Myopia is nearsightedness, and it can be corrected by the help of concave lens. ठीक है? Okay. Yes. class 10th physics. Okay, now it's time to apply it. So this option P2B, so P2 but B, so ये तो गलत हो गया आपका, ठीक है. Q1B is correct, of course it is correct. Q1B, P1A, again it is wrong, and this is also wrong. Correct answer is B. Very good. Okay, let's move to the last question of this paper. Now this says, which one of the following properties causes plant tendrils to coil around a bamboo stick? Okay. Okay. तो ऐसा बोल रहा है कि मान लो कि एक प्लांट का टेंड्रिल है, ओके, और यहाँ पे एक बैम्बू स्टिक है. सपोज दिस इज द bamboo stick, okay, and it is saying that when they have the tendril goes and coils around it, right? It coils around it. The question is, what makes this tendril to coil around this bamboo stick? Okay. Okay. देखो, without a lot of, wait, let me tell you, the option C is correct here. Why? Tendril has spines. It is not so. The base of tendril grows faster than tip. If the base will grow faster, obviously it will ultimately lead to the increase in the length of the tendril. But it can't lead to coiling of the tendril, right? Part of the tendril in contact with bamboo stick grows at a slower rate than the part away from it. Okay, इसके बारे में हम फिर से बात करेंगे. D, the tip of the tendril grows faster than the base. अगर टिप तेजी से कर रहा है उससे क्या फर्क पड़ रहा है? It can grow in a straight line, that will not cause any coiling. तो ये सब तो ऑप्शन होगा ही नहीं, राइट? ऑप्शन सी ही हो सकता है ना. कैसे होता है वो मैं आपको समझा देता हूँ. फॉर एनीथिंग टू रैप अराउंड, कैसे होगा? मान लो कि आप एक बाई मेटालिक स्ट्रिप पढ़ते हो ना फिजिक्स में, बाई मेटालिक स्ट्रिप, राइट? इफ मान लो कि एक ऐसा बाई मेटालिक स्ट्रिप की तरह ट्रीट कर लो किसको? अपने टेंड्रिल को. सपोज दिस इज योर टेंड्रिल, okay? Now it has clearly two sides. I am taking two sides here. One side I am painting in yellow and the other in red, okay? Now if I say the yellow is growing, the yellow side is growing at a slower speed and the red at a faster speed, after some time what will happen? In the same given time, this will grow this big, but the yellow in the same time will grow only up to this, right? Because of this, this whole plant have to move on what? On the yellow side. समझ में आ रहा है? तो जो साइड कम ग्रो करेगा, बेंडिंग उसी साइड पे होगी. जो ज्यादा ग्रो करेगा, बेंडिंग उसके ऑपोजिट साइड होगी, राइट? यू कैन अंडरस्टैंड दैट, ओके? सो ऑलवेज जहां पे भी हम लोग इस तरह का कर्वेचर के अराउंड मूवमेंट देखते हैं, इट इज ऑलवेज टुवर्ड्स द साइड वेयर द ग्रोथ इज स्लोअर. Now why slower? It is slower because of the degradation of auxine, right? क्यों? इधर जो है on the yellower side there will be low auxine, okay? And on the red side there will be more auxine, right? Auxine is responsible for plant growth. More auxine means more growth, so the plant will move on the other side. On which side? Where there is less auxine, there will be less growth and there will be the curvature. This is the same mechanism that you have studied for the phototropic movement, correct? बस यहाँ पे response stimulus किससे मिलता है? Touch से. इसीलिए हम इसको क्या बोलते हैं? Thigmonastic movement, right? We have already discussed that in another question, in the previous questions, correct? समझ में आ रहा है ये चीज? Let me comment and let me know कि ये समझ में आया कि नहीं आया आप लोगों को. Okay, देखो. So this is it. थोड़ा सा मैं आप लोग को ऑक्सीन के बारे में बात कर लेता हूँ. See, when we are talking about auxine, auxine is secreted from the apical part, okay, from the meristematic parts, primarily the root and shoot apex, okay, and this is responsible for apical dominance. ठीक है, apical dominance. When we are talking about gibberellin, this is responsible for bolting effect. Bolting effect, that is elongation of internode, या internodal elongation लिख देता हूं मैं, है ना? In just a minute. Auxine for apical dominance. Gibberellin for bolting effect, है ना? और ये किसकी वजह से होता है? Because of internodal elongation. Okay. When about cytokinin, overall the branching of plant, है ना? That is antagonistic to apical dominance, है ना? Apical dominance के इफेक्ट को ये कम करता है. कौन? साइटोकाइनिन. What about ethylene? It helps in fruit ripening, है ना? ये सब थोड़ा थोड़ा कुछ important है जो कि आप लोग याद रखेंगे. And when we talk about abscisic acid, okay, that is also called ABA, it is responsible for abscission, that is falling of leaf, of leaves, flowers, fruits, etc. है ना? Matured flowers and fruits. Okay, great. Yeah, okay, so this is about most of the all the 5 plant hormones. There are only 5 plant hormones, okay, out of which the only ethylene is the gaseous one. And all of these phytohormones are extensively used in the agriculture. Okay, so this is all. We have covered the 20 questions, all the biology section of 2018 paper. Now, I hope you enjoyed it, and thank you very much. We will meet again in the next theory lecture, probably this week. Great. And in that class, we will be discussing about the human physiology. Okay. Okay. So thank you. I'm signing off. Thank you for being with me.